what should you consider when choosing which method to use for your biologics project? Relative to product protein, method A has some quite specific advantages. Overall, it's applicable to all host cell proteins in the sample, so it's not bound to any specific host cell protein, which makes it quite desirable. It normalizes for sample preparation variability. The drug product is present throughout the entire sample preparation process, so it will be under the same conditions as your host cell proteins. That accounts for some of the sample preparation variability. The sample setup is quite fast, making it easy for fast exploratory analysis. However, it is only suitable for early discovery, which would be one of the downsides of this method. On the limitation side, there's a large dynamic range between the product protein, but also the HCPs, so that can cause some issues. It's difficult to compare across different sample types and product proteins because, as you saw in the earlier figures with the precision, for example, depending on the reference standard and method used, you will get different results. So if you want to look across some different projects or different biologics, this would not be possible with this method. Unexpected modifications in the product protein can affect results. So if the peptides you're using somehow get affected or have a modification, this will and can affect your results. For method B, relative to spiked-in protein, it is also applicable to all host cell proteins in the sample. It also normalizes for sample preparation setup as it is spiked in prior to digestion. The sample setup is quite simple as it's with known spike-ins. This method is suitable for process and product characterization. It also supports the transition to a target MRM approach, which is one way it discriminates itself from or stands out from method A, which due to the high dynamic range of the product protein cannot be used for a targeted approach. It enables a universal method where you could have the potential for a generic reference protein because you would be able to use it across different projects as it is spiked in. There are some limitations, however, as well. It also requires a spike-in process, but also comparison with and without spike-in to ensure there's no inference. The untargeted method has a slightly higher lower limit of quantitation compared to the targeted method when using relative to spiked-in protein method. Method C, relative to spiked-in peptide. This is only suitable for targeted approaches. It is effective for specific host cell proteins of interest. So if you know you have some high HCPs of concern, monitoring them with this is an effective way. For example, clustrin or light LPL. Pre-made isotopically labeled peptides are available for some known host cell proteins of concern, again, like clustrin and LPL. But there are some limitations with this method as well. For example, it relies on complete proteolytic digestion of the peptide, recovery of host cell proteins. Because it assumes that there's a complete proteolytic digestion of the host cell proteins, the results may not always be comparable because it depends on how effective your digestion has occurred. Spike-in timing pre or post digestion will also cause variable peptide losses. Synthesis of peptide stability may also vary, so depending on the peptide you have and the synthesis stability of this, your results will also vary over time.